So I can introduce our speaker today. Dominik is a postdoc in SETI Lab, right?
Correct. Yeah.
SETI is the name, right? Perfect. He received 2 degrees in mathematics from Berlin, where he specialized in topology and dynamical systems. And today he's going to be talking about Mellon, which is a non-parametric cell state density estimator based on a nearest neighbor distance distribution. I'm pretty sure you will say it way better than I did. I just read the title.
Absolutely fine.
So, a small request from Dominik is that feel free to just either raise your hand or write on chat or just jump in, and he can take the discussions and questions either in the middle or afterwards. So, Dominik, you can share your slides, and I can confirm that we can see everything. The floor is yours.
Right. There you go. I hope you can see it all right.
Perfect. Everything perfect.
Yeah, so I want to talk about Mellon, our cell state density estimation tool. And before I jump in, I just want to state the problem that we are working with here. So in our lab, we often look at hematopoietic stem cells from either mouse or human. And this is a system of differentiating cell states, often viewed in this discrete way here, where we have some stem cells that then undergo different intermediate states or state transitions all the way into some blood cells like erythroids, monocytes, granulocytes, and so on and so forth. And the nature of the system is also that this is a somewhat continuous process where cells don't just flick a switch and they become the next intermediate state, but there is continuous transitions. And this also manifests in the data. And that makes it, let's say, these challenging to work with discretizations, with clustering, and just grouping those cells and applying our approaches that we often do in other data sets to this continuous system. So a common way of viewing or perceiving this kind of process of differentiation is through something called a Waddington landscape, which is thought of like an energy potential or a differentiation potential that cells roll down on, just like a ball or water would flow down a hill through all these valleys. And the shape of this landscape thought of to dictating this process. So it's an abstraction how we could view the process and how the underlying mechanisms guide the cells down different pathways. And what we often see when we look at the distribution of cell states is that cells are not uniformly distributed along their pathways. There are certain accumulation points and very sparsely observed cell states in between. Now, when we look at a real data set, like here, that's a UMAP of T-cell depleted human bone marrow, we see this kind of branchy structure. There's often there's of course a stem cell compartment down here. And then there's all these branches into different cell types. And the one that's highlighted here is the trajectory all the way into naive B cells within the bone marrow. And you can already see how connected all this is. And there's no clear or no natural cluster boundary. And how can we look at this kind of data? Now, one common approach that we first do is to compute something called pseudotime. So this gives the cells an order in their temporal progression. We don't know if this is actually how fast the actual time is progressing. It just indicates which cells come first and what the intermediate states are and how to sort these cells if you want to look at something like gene expression development and so on. And here's, if you depict this for only the B cell trajectory, we can already see that there's certain accumulation for known cell types, the described ones, which are more common and something much more rare in between. But it's not quite clear from this picture if this is actually real or if this is just an artifact of how pseudotime is computed. But this is kind of the motivation of looking into this more deeply and maybe finding something else and just annotating clusters and combining cells into clusters to understand the process. So when we want to look at this density more directly, some people may turn to computing the density of cell states directly on the UMAP because it's a cell state representation. And some people often hope that this is a meaningful distribution of cells. But we know that this is not guaranteed to be true for UMAPs. And we also know that the two-dimensional representations that we have here don't contain all the biological variability that really exists in the system. And computing a UMAP slightly different can yield a very different result. Let's say we change the parameters or the number of nearest neighbors, and the two densities don't look alike that much. So it's not really a stable result, and it's also not a really detailed result. And we don't have any, let's say, mathematical guarantees that this is actually meaningful biologically. So something else that people often turn to in order to get a better grasp of this density distribution is to compute nearest neighbor distances between cells in their respective cell state space. But this is also very noisy, and it's really hard to read something into this biologically. So our idea was to do something to get a better density function, to get a better grasp of the underlying processes and what are real accumulation points, what are real transitory states, and several other applications that I can allude to a little bit. So the problem is, how do we compute this kind of density in a more meaningful way? So we turned again to the distances between cells. And what we use in this algorithm is mostly the Poisson distribution, and the idea that we can compute the probability for a neighboring cell to be within a small sphere around a cell based on the density. And this probability is just the probability for a cell to exist there. And this can be formulated as an integral. And we can use this formulation to get a density function with respect to the radius around a given cell. And this density function essentially represents a nearest neighbor distance distribution. And we can use this to link stochastically or in probability the density at a given cell state with the nearest neighbor distance that we observe. And we can use this in a Bayesian network or in a Bayesian inference model, where we formulate this connection between the density and the observed data, which is the nearest neighbor distance, through this distribution. So the nearest neighbor distance distribution only depends on the dimensionality of the space and the density at this location. And because the density is not a constant thing throughout the whole space, I mean, this is the interesting part, right? The density varies. We treat this density function as a parameter in our Bayesian model. And this density function, or the log density function, is only restrained through a prior, which is given through a Gaussian process. And the beauty of this is that this allows us to sample functions quite generally, but only restrain the smoothness of the function. And by restraining the smoothness of the function, we essentially link the density that has to come out between neighboring cell states, because the function only changes at a very low rate. For instance, the density between two neighboring cells has to be somewhat similar. So the density function that comes out has to explain the density or the nearest neighbor distances for multiple cells which are in close proximity. And so we can increase our statistical power, and by only using the assumption that density between similar cells is similar, which we believe is a reasonable assumption. So we have this Bayesian model formulation, and we can use this for any inference approach that we want. And we hope to get a grasp on these processes which impact the density. So for instance, we have proliferation, of course, where we would see an accumulation or an increase of cell density. We have apoptosis along this differentiation, where density is expected to go down. But we also often have acceleration of transcriptional changes, or just cell state changes in general, however the cell state is measured. And we also have deacceleration, where cells might accumulate into a more stable, more slowly changing cell state. We could also have convergence, where cells all go through a very narrow path in the cell state space, or divergence, where they all change their state more randomly, which is of course not directly expected in biology in many cases, but it's something that that can be observed by looking at density, we hope. So this is how this would look like for this toy example that I drew here, where we have those high and low density areas. And this is how it would look like, how it looks like on the real data set I showed you before. It's a slightly different UMAP, but it's essentially the same data. We have the stem cell state, and we have this progression into naive B cells. And this is what comes out of Mellon. It's the log density, essentially. And you can already see certain like high density areas and the low density transitions in between. And when we look at these cells over pseudo time and look at their density, we already see that the peaks in density are associated to the described checkpoint states of the B cell. Those are the cell states being observed. But we also have all these transitory cells in between. And when we really look at these transitions on what's happening there, we, for instance, already see that based on fate probabilities that we can compute with Palantir, that this is, for instance, the commitment phase, where the fate probability really goes up. And when we look at what's happening there in terms of gene expressions, for instance, one of the main transcription factors of B-cell differentiation really comes on in this transitory phase. And our idea was to find a way to detect all the genes or all the mechanisms that play a role in this transition. And to get a grasp of this, we compute something called local variability, which is only based on the difference in expression between all the neighboring cells in the nearest neighbor graph. And by using this variability and weighing this with 1 over the density, we can pick out all the, let's say, most rapidly changing genes that might facilitate this transition from one cell state or from one major cell state to the next one. And we pick out EBF1 and some other important transcription factors, but also some new stuff that seems to be interesting. And yeah, this is also implemented in Palantir using the Mellon density output. Also using Mellon is implemented in Palantir. So you can just, if you're already familiar with the tool, call it through Palantir to compute the cell state density on your data. And why is this interesting? We believe that cells make a transition from a high density state in a low density state, because we know they progress through the whole system. There's only certain explanations that can explain in a mathematical model how this density drops comes to pass. So one explanation would of course be rapid apoptosis, that all cells progress at an equal velocity through the state space, but at some point many cells just die. But the density differences are very severe, and that would mean that 99% of the cells are dying at a certain point, which we can often rule out as an explanation in the system because it would be biologically very ineffective. Another explanation for a drop in density could be divergence, that cells just spread out into all directions of state space, which of course also doesn't really make sense because it seems to be a very regulated mechanisms that keep cells on a very narrow path from one major cell state to the next one. So the most likely explanation often is that this is happening through an acceleration in their transcriptional state changes. So that there's actually some trigger that's happening right there at the transition from the high density into the low density that starts a cascade of transcriptional changes that happens in fairly rapid progression until they hit the next stable cell state. So understanding this process might really help us to understand how this progression is triggered and what the mechanisms are driving the cell differentiation. And to look at this through a different lens, we can come back to this idea of a Waddington landscape, which is an energy potential or perceived energy potential to explain this differentiation of cell states. Because if we assume that this energy potential induces a Boltzmann distribution, then we have a direct link between the density that we are computing, here as a toy example on the left, and the energy potential, which is a Waddington landscape here on the right. Of course, we know that the Boltzmann distribution doesn't include any form of proliferation or apoptosis of the states that are being included there. And this, of course, plays a role in cell differentiation systems. So there is no direct one-to-one link here between the density and the Waddington landscape, but at least on a very local scale, where the proliferation or apoptosis rates are approximately constant, there is an association that might be useful if somebody wants to look at the Waddington landscape more directly. And so the way we are computing our density function in the Bayesian inference model I showed you before, we are actually computing a continuous density function. So this function can actually be evaluated in between cell states, and you can also compute gradients and derivatives of this function in order to analyze it further. So one thing I want to point out here in this talk is the stability of the approach. We did a lot of benchmarking, varying many parameters. Here we only vary the number of cells being included. So we would downsample the full data set, which is here on the top right, with 10% at a time, reducing the cells, recomputing the density, the log density, and then looking at the Spearman correlations between the two density functions, just to show that this is a very stable result, even under substantial downsampling. Of course, Spearman correlation isn't a perfect analysis of how consistent the results is. Consistency results are, you might want to look at more subtle details. But at least on this global level, it seems fairly stable and robust. We also looked at the scalability of this approach. So first up, here's an example of the runtime this algorithm needs for 250,000 cells. And we split up the runtimes in several steps, including the pre-processing steps, which are PCA, and the diffusion maps, which we use to represent cell states. Of course, the diffusion maps aren't required. You can use any cell state representation that you would like. There's also no constraint on the dimensionality of this representation. The main importance lies in how meaningful the Euclidean distance is between cells in your cell state space. Ideally, this Euclidean distance and the representation mimics the biological dissimilarity. So it has some biological meaning of how much work it takes from one cell state to the next one, because the cell state will be formulated on this space, and closeness in the space should mean biological similarity for this density to be meaningful. Also, because we implement a sparse Gaussian process for scalability, we use some landmark or inducing points in our state space. And by default, we just use k-mean centroids, but this can also be swapped out for anything you like Leiden clusters or any other good subset of your data set. And these landmark points are used to formulate the density function. So the more you have, the more detail you get out of the function. But we showed that the default values we are using here, like 5,000 landmark points, suffice to fully describe the density function, again, by looking at Spearman correlation between the full density function and the sparse version for different landmark points. And it's all in the paper. And we think this is a good approach. We also have some mechanism or a part of the algorithm that actually evaluates if you're exhausting the complexity of the function based on these landmark points. So if you happen to use too little landmarks, Mellon will actually give you a warning indicating that you might have to increase the number of landmarks to get the best possible density function. Then we have this matrix decomposition, which is used for the internal representation of the density function. And there's a quick initialization and then the optimization. By default, we are just optimizing the posterior function. We're just finding the one density that maximizes the posterior. But you can use other optimization approaches as well. For instance, we also implemented automatic differential variation inference, where you can get the mean of the posterior density distribution and the standard deviations. And we also gave access to the posterior function itself, together with the initialization value, so you can actually use any optimizer you want if you prefer something else in the algorithm. Now, in terms of like scalability to data sets, we subsampled a bunch of different data sets to look at the runtime for them. This is on log, and the diagonal here is the actual diagonal. So if we comply to this, it means there's a linear dependency. And if we have less cells than those 5,000 landmarks, we are using the full Gaussian process. And it's of course not quite linear, but as soon as we hit the number of landmarks, the approach or the runtime increases approximately linearly with the number of cells that we are putting in. And we also showed that it still works for really large data sets. So we just simulated 1 million and 6 million cells up here, but we used a reduced number of landmarks to actually fit into memory. And it still works. So it's not exactly linear, but the other nonlinear terms are not dominating this runtime here. This is only the single core performance. And here on the right, you can see if you use multiple cores, because it's implemented in JAX, you gain actually some performance in the algorithm. And of course, if your data set is small enough to completely fit into GPU, you can use JAX to just run it on GPU and get even faster results. So just a quick overview of like how the internals on Mellon can be controlled. You don't have to set any of these parameters if you would just want to run it out of the box. But we try to give access to any internals you could wish for. You can specify the optimizer here. This is where you would pass any different set of landmarks, for instance. And you can also, for the Gaussian process, who's familiar with it, change the kernel function to control the smoothness of the outcoming density functions even more directly. And how would we use this with NData? I think it's fairly simple right now. We initialize our model, get some representation, whatever we like. You then just fit the model on it. It does everything you need for you. And then you can extract the predictor. You can, of course, just call predict directly, but the predictor is actually a separate object. And by just taking the predictor, we shed away everything else that's needed for the inference. And this is actually the part you might want to work with and you might want to keep. You can then, of course, evaluate the density on any cells. That can be a subset, that can be a superset, but of course, it has to be in the same state space to get the log density function. And you can serialize it fairly easy. So there's also a 2JSON version, but this one is a 2 dictionary, which only gives you a dictionary of strings, hierarchical dictionary, and putting it into the NData like this allows you to save it without any problems. And of course, you can then take the serialization and get it back to its predictor class for later evaluation. And of course, you can have any other cells that you want to evaluate the predictor on, but they have to be in the exact same cell state space. Otherwise, it doesn't really make sense. And this is not quite everything that we can do. We can also look at time series data. So here we have an example where mouse guest relation was sampled throughout multiple time points of the embryogenesis. And we can, of course, compute the density on all these subsets. But one thing is that this is a continuous function. So we can evaluate this density even on the cell states which were not included in the time point, which can already be helpful to make comparisons from time point to time point. Because now we have 2 density values for each cell state, and we can compare the correlation or the differences that are happening across time, even though we didn't include the same cell state in both of the samples up here. But we can also actually make this density function continuous in time, because that's just another access to our space. And of course, we use a different covariance for that. It can be controlled separately. And if we do this, we get a nice time continuous density, which is shown here on the right. Down here, you just see how we scroll through time, essentially. And we can look at all the interpolated intermediate density values. And because it's differentiable, we also get the time derivative here on the left to see where the density is increasing at a certain time point or where it is decreasing. So the same thing, if we want to do this for NData, there's only very minor changes. Of course, we now need some numerical time representation. And when we initialize our density estimator, we might want to specify how quickly the density is changing over time. So this is how smooth the density is across time. There is also an internal way to infer this automatically, which is fairly costly by it does leave one out computation of all the possible densities and looks at the correlation to infer how quickly it is changing over time. But it's often enough to just apply a heuristic in most data sets, which we also explain in our tutorials. But the more general approach would, of course, to just leave it out, but it takes a lot longer. So then, of course, we have to specify the time, but the rest just stays the same. And of course, we can evaluate the prediction at any specific other time that we like, and we can get the time derivative out of it. Yeah, but the rest is essentially the same. And one last thing that I want to highlight here is because we implemented or we put so much work into implementing the Gaussian process, the sparse Gaussian process, and making it really effective for biological data to infer these smooth functions. We also give access directly to the function estimator to the Gaussian process to use it for other applications. For instance, if you have some X data, like the diffusion map eigenvectors, which we use to compute the density value, and you have some function values for this data, like the gene expression, you can actually get a really fast function estimation to give you a smooth function for whichever function values you're interested in, like the gene expression, which would be a very canonical example, where we just use the very noisy expression that we measure in our single cell data to train the smooth function, which essentially can act like an imputation of the gene expression, and it's fairly quick. We can also do this for any other quantity that we like. For instance, we can make a pseudo time function. You might say, okay, we already have a pseudo time for all the cells, but there might be benefits that when you compute your pseudo time, you don't want to include all the cell states because they mess up your nearest neighbor graph or something like this. So you just use a subset to get pseudo time, but then you want to evaluate the pseudo time for the cells that you excluded. So this way you have a continuous function which you can then apply onto additional cell states that were not included in the pseudo time computation. And the last thing is, because it's differentiable, you can also compute the gradient of any function that you want to get out of. So for instance, if you have a pseudo time, you can compute the pseudo time gradient, which gives you some directionality of movement based on pseudo time. And you can do this for many other functions that you would like to evaluate this way. Yeah, so I just wanted to highlight this point too. So we have our paper as a preprint out right now on bioRxiv. The code is on GitHub, and we are waiting for the paper to be published in Nature Methods. And with this, I want to thank my lab, the SETI lab, and all the great support. We have developed this tool together, and yeah, we are very happy, and now I'm ready to take questions.
Perfect. Thank you so much. And congratulations for the coming publication. I hope it goes as smooth as it can be, this process. But, you know, it sounds like you've been through most of it for now. So any questions, you can raise your hand, just open the microphone, whatever you want. It can be discussion even, or write on chat and I can read it for you if you don't want to. So usually people are not shy, but give them some time. So I can start then. Usually I don't get the time to ask. I have some questions, but it's a bit kind of in the beginning that when you start presenting. So you mentioned at some point that with Mellon, you did some analysis that it was already kind of done with the hematopoietic cells, I think. And then you said that you found concepts that they were there, like housekeeping or something expected, but also new ones. Mm-hmm. So I'm very curious, what was the new ones? I guess I can guess that.
So honestly, we haven't looked at all the candidates we found out there in detail yet, but I think some of the most interesting ones were those associated to cell-cell communication and cell adhesion, which makes a lot of sense for the process of B-cell differentiation as they migrate out of the niche and adhere to maybe a different location. So yeah, we had like this. Sure, I think I showed like 2 or 3 cells on the slide too.
Mm-hmm.
oh, it's here, right? So we had this, it's like a neural regulator for a cell adhesion, which comes up really high up here than this DIAPH3, which is also associated to cell migration. And I think those would be really interesting to investigate. But of course, we are a dry lab, so we rely on collaboration to really look into this.
Yeah, that I get. But so you are at the point that you could with Mellon suggest some candidates, for example, and narrow down to few ones so they can actually go to the wet lab, right?
Right, exactly. Yeah, and this is this should be fairly accessible to you can put in like any transition you want and then apply this just to get like a list of genes out that are changing. And then of course I would always recommend to really look at the genes and their expression either on the UMAP or across pseudotime to see if it really makes sense, to see if the expression values really make sense. But for us, it already helped to narrow down the list substantially, and the top candidates were really interesting. And yeah, we are hoping in collaboration to find out more about this.
Okay, that would be super interesting. Let's see if there are other questions. Keep going.
I actually have a question on the same slide. In this graph that shows the log density over time, in this low density region, you have some quite large jumps between whatever cells or landmarks, what a dot represents. Is this a technical artifact or is this actually a signal? How can you explain this?
What do you mean by chunks? You mean the amount of cells?
Jumps between, but in the low density region, there is a point that is halfway on the axis and then the adjacent one above.
These?
No, further to the left, basically.
These?
So you have one dot, and then the next one is basically much further down, then it goes up again.
Okay. You mean while there is so much noise, essentially?
Yeah, exactly.
Of course, the cell state space is fairly high dimensional. I think here we use a 20-dimensional representation. So we are just projecting down to this one-dimensional pseudotime. And of course, there's a trunk of cells along the pseudotime. But if you go to the left or the right, orthogonally to this pseudotime, density, of course, also drops off. And if there are cells which are to the left or the right of this trunk, essentially, then they are in a lower density cell state because it's less likely for a cell to be this far away from the trunk, essentially. Yeah. This is the reason for this, essentially, that the density is computed in a high dimensional representation of cell state and not just on the pseudotime.
Okay. And then on the slide with just before the runtime evaluations, you showed that you were using diffusion maps as a low dimensional embedding before you go into the smoothing and applying your algorithm. Why of all low dimensional embeddings did you choose diffusion maps and not PCA directly or SCBI embeddings?
Yeah, so you can of course use whatever you think is most meaningful. We found the diffusion maps to be they're yet another dimensional reduction that improves the cell distances, and the diffusion maps seem to preserve the structure of the differentiation tree very well. In diffusion map space, you can see the branches literally that show the differentiation from the stem cells down to a more differentiated cell state. And it's a deterministic approach to get the diffusion maps out. And there is a mathematically somewhat rigorous reasoning what the meaning is of these diffusion maps and what the meaning of the Euclidean distance is in diffusion maps. So based on the theory, ideally the Euclidean distance in diffusion maps represents the diffusion distance, which in turn is explained through the different paths that a cell can take between nearest neighbor to get from one state to the next one. And if there is a lot of connectivity, essentially, then the diffusion distance becomes smaller because there seems to be a higher, let's say, likelihood for cells to have a transition between those two cells. And we found this to be meaningful. And we also looking just by playing around to yield really good results. And PCA often looks a lot more noisy and not as easy interpretable. But we also applied it to SCVI representations, which also look really nice. It's just doesn't have the same guarantee for the distance to be biologically meaningful.
Okay, thanks.
Mm.
Severin?
Hey, yeah, thank you for the really interesting talk. I have one question. You mentioned that you use JAX for the thing, and that you also allow for GPU computing if the data set is not too huge. I do a lot with like QPy. And do you also use JAX for the diffusion map? And do you allow this for GPU computing as well?
The diffusion maps right now are computed with Palantir on our end, which doesn't use JAX at the moment.
Okay.
So we treat this as a pre-processing step.
Mm-hmm.
But Mellon only starts at this representation. You basically feed in, right? Yeah, I had it like on this next slide here. So you give it the representation that you choose. So you can use whatever tool you want for the computation. And if you could make it a complete JAX pipeline if you want. Yeah.
Yeah. No, okay. I was hoping that you could tell me how I could fix the reproducibility with my GPU diffusion map. So there was.
Sorry. Yeah. No.
Okay.
Yeah. Yeah,
That's
maybe.
a good topic for discussion, though, if you have any hints for Severin.
Yeah, it might be a hard problem. But yeah, we can look at like maybe at some point we manage to use JAX and Palantir directly. But I don't know if it's going to be more reproducible. Yeah.
Good.
I have another question, if I may.
Yeah, of course.
I really liked your last example where you showed the how the density develops during embryogenesis, and that made me wonder if you could also use this tool for a comparison between conditions, like if you had a clinical trial and whatever treatment versus control or disease versus healthy, and you compute the density of cell states, can you then also compare them in a statistically meaningful way to say that there are changes in cell density for a certain state?
Yeah, absolutely. This is in fact the thing that we are looking at right now, the closest to make a meaningful comparison between cell state abundance from one condition to another. And we think that this could be a really valuable approach to using density directly like this. There are already abundance difference detection tools, but Mellon allows you to get an absolute quantification of these abundances. And it also gives you a degree of uncertainty or a measure of uncertainty. You can, in fact, compute based on the Bayesian inference using variation inference, the uncertainty of your parameters and translate this into the uncertainty of the density values, which in turn then can be used to give you a degree of significance of any density difference. And so we hope this is going to help to detect more reliable where there are meaningful density or cell abundance changes from one condition to another, from old to young and stuff like this. Yeah, absolutely.
You're also not relying on discrete cell types here, which I think is a limitation of all the other tools that compare cell type abundances.
Yeah. We don't have to use any discretization, and you can evaluate this difference of abundance at any arbitrary point. I think where you have the most data, the comparison is going to be the easiest. When we quantify this uncertainty, we use both the source that comes from the Bayesian inference, but also the source that comes from the density representation, because the density function, it's a function, right? So it's infinitely dimensional. We cannot store the whole function in memory. We have to rely on a low dimensional abstraction, and this results in an uncertainty of the function values when we get too far away from the data. But yeah, we certainly don't have to reduce the single cell resolution to do this. And there's other tools which also do a great job on this, like MELT, for instance, that gives you a per cell density abundance difference. But yeah, we can even go, I think, beyond this by evaluating it in between, for instance, or like bringing in new data and then computing the density on this and comparing between multiple conditions. Thank you.
More questions or comments from your side? What is the future now? Like, okay, publication first, I guess, and clinical trials.
A lot of things that build up on density, essentially. There are multiple ideas. One, of course, is a differential abundance thing that hopefully comes out soon. But I also want to get a better grasp at the differentiation process itself using this density, and maybe using the time continuous density, but even the static one can be helpful to inform any approach to infer the differentiation mechanism.
Yeah, sounds really good.
Thanks.
I don't see any more questions. People are leaving, so I would like to thank you before everyone leaves. Thank you for joining and presenting, and everyone can reach out to you. I don't know if you're on Zulip, but you should definitely join on Zulip. I shared, yes, I can text you later on the email. Yeah, and thanks everyone for joining. See you in 2 weeks.
Thank you very much for having me. It was a great pleasure. Hopefully see you soon. Bye.
Bye.
Bye.